Both of these are powders of lactic acid bacteria that have been cultivated and heat processed. The lactic acid bacteria on the right has been nanoprocessed, while the bacteria on the left has not been processed. Both are dissolved in water. This image shows stir a water suspension nanoprocessing lactic acid bacteria and become white to turbidity. This image was taken using a microscope. The particle of nanoprocessing lactic acid bacteria is smaller than 1 micrometer. On the other hand, the particle of no nanoprocessing lactic acid bacteria is larger than 20 micrometers. No nanoprocessing lactic acid bacteria is stained the large red clumps. The clumps of no nanoprocessing lactic acid bacteria are not broken down into dispersion even after treating them in artificial gastric acid for 3 hours and in pancreatin for 9 hours. They are both administered to mice to compare the way they are actually absorbed into the body. This is inside of the small intestine. The Peyer's patch is scattered here. Peyer's patches are checkpoints that identify foreign substances and microorganisms that enter the intestine with food. Switching to the fluorescence microscope shows the unprocessed lactic acid bacteria gathered around the edge of the Peyer's patch in large 20 micrometer clumps. Magnification reveals blurry images of clumps floating on mucus. This is after nanoprocessing lactic acid bacteria under a fluorescent microscope. The bacteria which dispersed in less than 1 micrometer is stained red. They can be seen firmly attached to the surface of the Peyer's patch where the blood can be seen flowing. Why is there this difference? The intestinal mucosa is coated in mucus at all times. The intestinal mucus blocked to no nanoprocessing lactic acid bacteria of the large clumps and bacteria can't reach the surface of the Peyer's patch. On the other hand, nanoprocessing lactic acid bacteria which dispersed can reach the surface of the Peyer's patch. It was confirmed where the Peyer's patch nanoprocessing lactic acid bacteria was reaching by confocal laser scanning microscope. This image scanned the Peyer's patch to the inside. The Peyer's patch and cells on the surface of the villi are shown in green, while the cell nuclei are blue and the lactic acid bacteria are seen in red. This image is magnified and a cross-section is examined. Many nanoprocessing lactic acid bacteria which dispersed are absorbed in the inside of the Peyer's patch. Various immune cells are staying on the inside of the Peyer's patch. Nanoprocessing lactic acid bacteria that are absorbed by M cells are passed on to dendritic cells and macrophages, which then activate T cells and B cells.